Okay, perfect. I love that. Yeah, if you would like to share your screen with us, please. And then Adriana is actually, her and I met when I moved to USA, actually, because I'm originally from Czech Republic and I moved to US about 3.5 years ago. And I met Adriana at a cat show. She is an amazing Tika judge. She actually was Tika Judge of the Year in 2013 and then again in 2022. And she is a fantastic educator. She really was the one that inspired that we do this conference. I'm really grateful to her because I think it wouldn't happen if she wasn't pushing me so much to organize this. So thank you. And also, Adriana is not just an amazing cat breeder and Tika judge, because she also breeds Singaporeans. She also is has a PhD in human virology, so she's a scientist as well. And I really have enjoyed all of our talks that we've had about cats, and I hope you guys are gonna enjoy this last talk of the day for today, because we're of course continuing with more education for you tomorrow. But Adriana's talk will be a little bit longer. It's actually 90 minutes because she wants to share a lot of stuff with us. And like I said, she's very passionate about breeders' education. Just a heads up, Adriana will make a break in about 45 minutes of her, like halfway through her talk, and actually accept questions halfway through, because this is a 90-minute talk and there will probably be a lot of questions because we are going to be talking about genetics of coat colors and patterns. And I've been a judge now for almost 10 years and breeder for even longer, and I still find some colors sometimes really challenging when we see them on cat shows and breed them. So I think there will be a lot of questions. So Adriana kindly agreed to make a little pause halfway through so we can clarify some things and answer any questions then. So thank you, everybody. And Adriana, the floor is yours, and I cannot wait to hear.
Well, thank you, Petra, for the invitation to be part of this amazing team of speakers in this first conference, hopefully first of many. Like all of us joining you in this adventure, we believe that it's through education that we are going to make a difference in the quality and health and overall quality of life of cats. So here we go. Brace up, everybody. This is probably dense content, but I hope that my approach to teaching genetics of coat color and pattern is something that will be appealing to you and a strategy that will help you understand the picture better. So you will be walked through different aspects that will have a major foundation on biology concepts. It is my take on teaching this kind of content that if you understand what it is that is under genetic control, what kind of biological mechanisms are under genetic control, you will understand processes better and have a deeper ability to decide what is really important, what is functional and what is cosmetic. So the main learning objective definitely is to help you acquire a deeper understanding of the complexity of the biology and genetic control of coloration in the domestic cat. This is definitely applicable to every single other mammal on planet Earth, but we will focus, of course, with illustrations and mechanisms or modes of inheritance of the different traits on the domestic cat. So I will give you a couple of introductory concepts. One that is very important to me because I struggle. You probably notice I have an accent. I'm a non-native speaker of English. Like Petra, I was born, raised, and educated in Argentina. And one of my first impressions, well, we need to adjust the use of terminology. So the definition of color, what is color? It's a visual attribute of things, objects, beings that results from how the light is reflected by these structures. Whereas coloration, this term denotes a much more complex concept, it refers to an array or arrangement of colors and tones, the coloring and or the markings. So it's a general global appearance of a complex structure that results from the reflection of light. So going to the cat context, for a cat like this one, a Russian blue, saying, using the term color is perhaps appropriate. So you would say, yes, this is a blue cat. But what about this? When I present you with a black smoke and white Maine Coon where even the pattern is visible, using the term color, in my opinion, is inappropriate. So you will hear me at shows, events, chats, dinners, and formal lectures like this one, referring or using the term coloration. So very important to situate the audience in the context of this talk. What kind of structures, what kind of tissues, what kind of cells are we going to talk about in the context of production of pigment and coloration? So many of you know some of these basic concepts. The cells responsible for the production of pigment in mammals are secretory cells. That means these are cells that secrete or expel out the content or the products they make, in this case, melanin or pigment, and these are secreted in the form of granules, and these cells are called melanocytes. The melanocytes derive from the neural crest. You can see in this little cartoon of the mammal, let's say cat embryo, at the top of the head. This is the site in the embryo where the melanocytes originate, develop, and start migrating from. This site is called the neural crest. And from here, these cells migrate and colonize different structures of the body. And this, of course, includes the skin and hair bulb, the eye. You will notice some critical areas of the central nervous system, including the medulla oblongata and, importantly, the inner ear. The pigments responsible for skin and coat color in mammals, again, these are called melanins, and there are two major forms of melanin secreted as granules by the melanocyte. Eumelanin, that is characterized because for the granules that have a round shape and they contain black or brown pigment, and pheomelanin, secreted in the form of irregular granules, and the pigment they contain is yellowish or reddish. The melanin granules are exported from the melanocyte onto the epidermis and hair bulb. So in the case of animals that have no hair or very little hair, such as us human beings, in most of our body we have very little hair. Think about hairless individuals, like hairless cats, like peterbalds or some sphinx, donskois. Where are those pigment producing cells? In the epidermis. The melanins, I don't expect you to cover the chemistry of melanin synthesis and production, but it is really important that you understand that these molecules are synthesized from a critical precursor, that is the amino acid tyrosine. And the very important thing that I hope you recognize from here is there are two divergent pathways for the synthesis of melanins. One pathway that leads to the synthesis of the dark pigments, eumelanins, and the other one to the yellowish reddish pigment, pheomelanins. The other fundamental aspect of melanin production that should be recognized from this slide alone is the connection with the production of neurotransmitters. In this case, we're going to stop here, just dopamine. So please acknowledge from now on that the production of pigment has a connection with level of activity, with behavior in many ways. And this is beautifully studied, beautifully published in mouse models, even in fox models, where there's a connection between differences in coloration and differences in pattern of activity and behavior. And what is the biological explanation for that? This particular biosynthetic pathway that has this initial step in common. For the actual teaching and delivery of content in this talk, I like to invite people to recognize four major, not yet there, but very important to recognize that the genetic control operates at the cellular level and at the organismal level. So inside the cell, different changes in the activity, connection, molecular communications, and the organismal level. In particular, we're going to look at how the migration of melanocytes that originate in the neural crest is orchestrated to colonize different areas of the body according to very specific programs under genetic control. Now, this is the way I invite you all to think about genetic control of coloration in the domestic cat and mammals. I like to group the genes in four major clusters based on the most important aspect that ideally you should try to incorporate into your toolbox as cat breeders and cat fanciers, based on their function. If you understand what is controlled by genes, you will be a lot more comfortable making decision on what is important and what is not important regarding coloration. So we recognize the first group, the group that I refer to as the group of master controllers. These are genes that regulate the production and the type of melanin produced. That means pheomelanin or eumelanin. A second group that regulates the maturation and distribution and migration of melanocytes. A third group that specifically encodes the enzymes that regulate the different steps in the production of melanin, the different players in that biosynthetic pathway that I just showed you. And a fourth group that regulates the shape of the melanin granules and their distribution in the hair shaft. We're going to discuss these four groups in the talk today, some with a little bit deeper level.
Please forgive me for interrupting.
Yes?
Can you click hide on your screen because we can't see the bottom portion of the slides
Oh,
because
thank you.
there's a notice.
Thank you. Is this better?
Yes, thank you.
Sorry about that. I saw it, but I didn't know what to do with it. Thank you so much. So this is group one. If you have to leave the talk, please stay for the duration of the slides that will describe this group. This is the group of master controllers of pigmentation. This is the group that we should be referring to and remembering as the pigment type switching system. So because it's a group, I am going to describe to you many genes, many coding regions in the genome that actually control not only the production, but the type of melanin that is produced. We need to go back to the cell, to the melanocyte, and understand how this activity is controlled. Imagine the melanocyte as a factory of pigment. So we're going to understand what makes this factory operate and how the operations of the factory are controlled. So a major controller is the melanocyte stimulating hormone, also known as melanocortin, depicted here as MSH, interacting with the receptor that is called MC1R, or melanocortin receptor 1, at the level of the cellular membrane. Very important to acknowledge that the production of melanin is a defense mechanism. Pigment production is required to protect any creature, any being from UV radiation that is mutagenic. So it has pathological effects if it gets through uncontrolled. So a first layer of protection, of course, is the hair. If there's no hair, the next level of protection is pigment. So pigment is biologically fundamental for protection of the structures of this organism from mutagenic effect of UV lights. And hopefully you will start thinking, and we can discuss at the end of the talk, the implications of being albino, of not having pigment, of being white. Topics for discussion. So again, fundamental concept, the production of pigment is a defense mechanism for protection from the mutagenic effect of UV light or UV radiation. So the orchestration of the activities of this factory are mainly under the control of two operators that compete for a place in the same receptor. One is the hormone, melanocyte stimulating hormone, or melanocortin. Acronym is MSH, and the symbol I will be using in the slides is this blue arrow here. The other one that many of you, I assume, have heard about is the agouti protein, or agouti signaling protein, ASIP, symbolized in the slides as an orange arrow. You can see that both the blue arrow and the red arrow can connect with a receptor. When the blue arrow is connected, the pigment that the factory will produce is the black one, eumelanin. When the red arrow is connected, when this ligand is connected, when the agouti signaling protein is connected, the cell will be programmed to produce pheomelanin. The factory will be producing red pigment. You will find me, you will hear me paraphrasing a lot, and this is to facilitate your understanding of these basic concepts. I do hope it works. So another depiction, another cartoon showing you the same picture, giving you a little bit of detail. So you will see the same receptor, melanocortin receptor 1, MC1R. You'll see two different ligands. Both activate the factory, but one activates the factory to produce red pigment. The other one activates the factory to produce black pigment. So variation in these key players definitely will be underlying a lot of the variation that we see in mammalian coat coloration. This is perhaps the easiest way for the majority of you to understand this absolutely critical level of control of pigment production. Imagine the ligands, agouti signaling protein, and the hormone MSH. Imagine those as two different keys that can be inserted in the same lock, the receptor, and open the sesame or activate the factory. So these are, by definition, these three critical players are the lead members or the top of the rank members of the pigment type switching system. Why? Because if the blue is connected, the pigment that will be produced is black. If the red ligand, acid, if this key is connected to the lock, the pigment that will be produced is going to be pheomelanin. So what do we know about the agouti locus? The agouti locus is the one that controls encodes the agouti signaling protein. And what we know about it is that it encodes a dominant form or a dominant variant of the gene produced in the dermal papilla cells. I will show you a cartoon of the dermal papilla in a few slides. And we know that this dominant protein competes the hormone, displaces the hormone, and when it is bound, the cell produces pheomelanin. This is the dominant form of the gene. We also know that the agouti locus produces a recessive form of the protein that cannot displace the hormone. So when this is the gene variant encoded in the genome of our cats, this is going to be a non-agouti individual. This is a protein that cannot displace the hormone, and therefore the individual will only be producing black, the dark pigment, black or brown pigment. And this is the animated cartoon. So we also know under the control of the dominant form of the gene, the agouti signaling protein. We also know, we've seen it in all our animals that we described as agouti cats or agouti individuals, agouti colorations. We also know that the hairs of these individuals are banded. They have bands of pheomelanin and bands of eumelanin in alternating appearance. And this hopefully is describing to you and depicting to you a picture where there's alternating on and off binding to the receptor and control of which melanin form is produced. Definitely the agouti coloration is the coloration of the wild. All of these colorations depicted in this beautiful picture, cheetahs, African servals, jaguarundis, lynx, jungle cats, tigers, all of these colorations are under the control of a dominant form of the agouti signaling protein. What do we know about the receptor or the lock? The receptor is encoded in the extension locus. Again, this is the critical player that binds these controllers that come from outside the melanocyte. The receptor controls the levels of an enzymatic activity that is critical for the production of pigment. This enzyme is called tyrosinase and is actually the first player in the biosynthetic pathway that I showed you. This is the enzyme that transforms the amino acid tyrosine in the next product in the pathway. So we also know that low levels of this activity result in the production of red pigment, and we know that high levels of tyrosinase activity result in the production of black pigment. A little bit more detail, and perhaps a more user-friendly paraphrasing of all these concepts. When the agouti signaling protein is bound to the receptor, preventing the interaction of the receptor with the hormone, the expression of melanogenic genes goes down, it is reduced, and the cell produces pheomelanin. So the branching of these two pathways that I was telling you about depends also on which controller, which key is bound to the receptor, which key is in the lock. Let's take a look at the effect of mutations in these two critical players, agouti signaling protein and melanocortin receptor 1. What do we know about coloration when these two critical players are different from the wild type? So for sure, I hope you can imagine already from what you've learned so far, that variation in these two critical players will definitely result in coat coloration variation or coat color phenotype variation. So what is melanism? Many of you have probably heard this term. Many of you maybe have melanistic cats. I'm sure many of you do. Melanism is by definition an increased amount of black or nearly black pigmentation. And I just showed you a picture of the look of the wild jaguars, jungle cats, lynx. Look at what I'm showing you now. I'm showing you melanistic coats in these different wild species. These are all melanistic coats. These are all darker coats that you will recognize as something different from what you call the wild type. So both ACIP and MC1R, when mutated, some of these mutations can be associated with melanism, can be associated with melanistic coat phenotypes in the Felidae, all Felidae, domestic cats or any of the big cats of the wild. Let's look at mutations in the agouti signaling protein. These two are some of the best known examples. Both the leopard and the Asian golden cats have mutants that result in melanism. And here they are. So this is the wild type agouti coloration in the leopard and the Asian golden cat. And over here you have the agouti mutants that produce melanistic individuals. Let's take a look at what we've seen so far, what we've elucidated so far in the domestic cat that result from mutations in the agouti signaling protein. This is something we have all lived and been very well aware of for decades. This is an agouti individual or wild type looking individual. And what is this? This is a recessive mutant, non-agouti oriental short hair in this case. And this is a melanistic cat. This is melanism in the domestic cat. You are all very familiar with the looks. Perhaps what you are not familiar with or have not stopped to think about until today is that this is melanism. What else do we know? Thanks to the revolutionary impact that the Bengal breed has had on the cat fancies of the world, we've been able to learn more about genetics of coat coloration in the domestic cat, genetic basis of coat colorations in the domestic cats. And this is a body of work that came from the laboratory of Leslie Lyons. This is the first report in 2014 of a unique and distinct, the genetic basis of a unique and distinct look of Bengals. And these are all melanistic Bengals. They are all characterized by the fact that in their genome, they carry the agouti signaling protein version inherited from the wild ancestor of the Bengal breed, and that is the Asian leopard cat. So this acronym designates agouti signaling protein and the variant that comes from Prelionaurus bengalensis, and that is the jungle cat. I have to stop to bring you, just for connections and to expand your ability to think more broadly about color. The laboratory mouse, Mus musculus, is perhaps the best studied mammal on planet Earth. And in the mouse, we do know, the biomedical sciences field has known about many variants, many alleles of the agouti signaling protein. And here you have them illustrated, a variety of colorations showing you the wild type, agouti, capital, dominant form of the agouti signaling protein. And over here, the melanistic colorations you can see resulting from two different variants, but recessive forms of the genes. You can also see some variations on the theme, some yellowish colors, white-bellied or white tummy colorations resulting from variations in agouti. And please acknowledge that in the mouse, again, some critical connections have been made already, associating variations in agouti with important metabolic diseases such as obesity. This is the first invitation for all of us as a community to think about changes in color, especially as a result of changes in these critical controllers, resulting in pathological conditions. So this is the first invitation I will present you with through my talk, to think carefully about what is pretty, what's cosmetic, and what is functional, or what is healthy, or what has pathological implications, even if we think it's cool. So in the mouse, more than 9 variants of agouti have been described, and it's extremely likely that the same number or similar number of variants exist in the domestic cat and other mammals. They have not been discovered yet. That doesn't mean they do not exist. In general, in genes that control critical aspects of biology, there's a lot of conservation across the phylogenies, at least in the world of mammals. Examples very quickly. I just want you to see things that you've probably seen a lot in nature. These different colorations in dogs are also the result of changes in the agouti signaling protein. And you see in this French bulldog, the dominant yellow. You see here the typical recessive black melanistic dog coloration. What do we know about mutations in the receptor and coloration and melanism in felids in general? Definitely the Yaguarundi is one of the examples, the wild type and the melanistic Yaguarundi. And the jaguar is the other example, the wild type on the left, the melanistic jaguar on the right. The jungle cat is the other example. This is Felis chaus. So another beautiful example of the wild type coloration and the melanistic form of the jungle cat. What about the domestic cat? Just in the same way I presented you with mutations in the domestic cat for the agouti signaling protein and the impact on coloration, I'm going to show you what happens when we change the receptor and what examples we've been presented so far as members of the cat fancy. One is this. This is the Chausi. This is the breed that my cat association, Tika, has recognized for championship, as a matter of fact. A breed derived from a domestic cat hybrid breed derived from crosses between jungle cat and domestic cat. And we have the two major colorations recognized, the black tick tabby and the grizzled. The grizzled is a melanistic Chausi. What about this one? This is one that we've known in the cat fancy for at least a couple of decades. This is the amber coloration in the Norwegian forest. This is the result of a mutation in the receptor in the log, a recessive mutation that results in a progressive shift or switch in the type of pigment that the melanocytes produce. When the amber cats are newborn, they have the coloration that is basically designed and very visible under the control of the major players. So this kitten in particular is born as a brown classic tabby. Over time, as the mutation expresses more efficiently, the kitten or the individual experiences a shift, a shift from black pigment producing cells to completely red pigment producing cells, going through these intermediate colorations that are many times confusing, especially if the cat in question is a female. Many people tend to recognize or get confused that these are brown torbies or agouti tortoiseshells, and this is not the case. So this is the biology and nature of the amber mutation discovered in the Norwegian forest. Again, it's a progressive transition from black producing pigment to red producing pigment. There are others. And one of the ones many of you may not be aware of, excuse me, is the russet coloration discovered in Burmese cats, another recessive mutation that results in this unique coloration in a cat that would otherwise be sable. And you can see how there is no trace of black anywhere in the body of this individual. Another example of mutations in the receptor, the copal mutation discovered for Corellian bobtails. You can see this is the way this is reported in the genetics literature, the sequence of the gene, the localization of the mutations, and of course, the look of the individuals. In this case, there seems to be some progressive disappearance of black pigment production capabilities in these cells to a complete reddish coloration in the adults. Importantly, under a different designation, carnelian, this was also reported by this group in St. Petersburg in Russia, essentially at the same time, 2019. You can see it very clearly. I am not clear. I cannot comment. I have not seen sequence data, but most likely these two designations are referring to the same type of mutation, again, in the receptor. So this is the way we look at the whole protein, the receptor, and where different mutations have been mapped. This is interesting because they have been mapped not only for domestic cats or jaguars or jungle cats, they have been mapped for dogs, they have been mapped for foxes, they have been mapped for goats, they have been mapped for different species of cattle. So very important that we are, again, reminded of the fact that this general control of pigment production biology is conserved across the phylogenies of mammals. This is very interesting work coming from the laboratory of Chris Kalin at Stanford University. This is a very nice elucidation of the genetic basis of coloration in Bengals, where we can see combinations of mutations both in the agouti signaling protein and the receptor at the same time, with this being the wild type having the agouti signaling protein and the receptor protein encoded by domestic cat genes, Here we have the combination of agouti signaling protein from the jungle cat and the receptor of domestic cat. And this is agouti signaling protein and receptor from the jungle cat. So this is new body of data, new body of information that is and will continue inviting us as a community to think about how to enhance our tools for genetic typing. This is visible, but this is not very easy to type. None of the tests available commercially can do this for us yet, only specialized research laboratories. So again, a summary: agouti signaling protein and the receptor are the most prominent members of the pigment type switching system. Again, these very simplified cartoon that shows us two keys or two ligands, agouti signaling protein, the hormone, and the lock or receptor as the major players. These are the most prominent, but there are other loci, there are other genes capable of controlling what type of pigment the melanocytes, the factories, produce. So very important to be invited to think in terms of a pigment type switching system. We spent decades as cat fanciers thinking of agouti and then thinking of the receptor, the extension locus. Now we are being rapidly obligated, invited to expand our toolbox and incorporate other players and think of a pigment type switching system. Several instruments in the orchestra, if you want, to play this fundamental music in coloration. Corin is the most recently identified new player in the pigment type switching system. I'm assuming that the majority of you have heard this term. For those of you who have never heard about it, you are going to be introduced to it in the next slide. So what is this protein corin that some of us have been hearing about a lot lately? Corin is a protein produced by the cells in the dermal papilla, this structure in the hair follicle, this one here. The cells in the dermal papilla produce this protein corin, and what does it do? Just think the melanocyte is a different cell type. It's here. So we are talking about cells in the dermal papilla, and these little brownish structures here that are depicted or exaggerated in many ways are the melanocytes, symbolized here in this squarish structure. So the cells in the dermal papilla produce this protein corin, and what does corin do? What is the function of this protein? Corin plays a very important role. Corin is the mission on Earth of this protein is to degrade the agouti signaling protein and thus enabling the production of eumelanin. So when corin doesn't work, when corin does not degrade the agouti signaling protein, remember this protein can displace the hormone. So if it's not degraded, the factory will only produce red, and you have it depicted here. When corin works, when corin degrades agouti, then the factory can produce black pigment. So the production of corin during the cycle of the hair follicle biology or genesis, so to speak, or development, corin is produced on and off. and that's why we have banded hairs. So when choline is there, the agouti protein will be degraded, and therefore there will be a band of black hair. When choline is not there, when choline is not being made by the dermal papilla cells, the agouti protein will not be degraded, and therefore the factory will make red pigment. I am bringing you some information with some illustrations that unfortunately we do not have available for cat hairs. This is what happens when this choline protein is active. You see banded hairs when this fundamental player in coloration is mutated. What we get to see is what is described as a wide band or broad band gene. If we could pull a hair from a choline mutant, we would see this wider band, and in the cartoon you see it here. Definitely a lot of work conducted in mice. As I mentioned to you a few minutes ago, the mouse is the best studied mammal on planet Earth in a number of territories relevant to biomedicine, and one, of course, is coloration. Interestingly, the first report on choline relevant to the world of cats or relevant to us in the cat fancy came from the Luo lab in China. And this is a report on a body of amazing research conducted on tigers. So again, just to remind you what we covered, essentially it all emerged from the work in the Luo lab in Beijing, elucidating the genetic basis of this dramatic change in coloration in the tiger. Of course, now we know it's attributed or the result of a mutation in the choline gene. So that results in hairs with broader bands, and this is the in the orange areas, but also the stripes, the pigment in the stripes does shift, and our eyes see it as a different tone, browner or reddish. Okay, again, then you missed it. In the domestic cat world, the first body of information came from research in Siberians that started coming out in some litters with these dramatic changes in coloration from a typical coloration brown classic tabby or mackerel. This shift where you can see a significant higher abundance of reddish tones in the coat. Even in the silver coats, we see the same dramatic effect from black brown tones to reddish orangish tones. And if we start plucking hairs and looking at them in the microscope, the common feature is that all of these hairs have broader bands where the pigment incorporated into the hair shaft is yellow red pigment. More examples with more mutations identified in the Siberian breed. Definitely, this is generically we can refer to as a wide band phenotype. These are called golden cats. And as we know, we all are very aware and have to work on this. We have to come up with some consensus terminology to designate these colorations so that all of us members of the cat fancy operating in different associations can speak a common language. The British Shorthair is also one of the breed populations where some choline mutations, different choline mutations have been found, and the phenotype, the coat coloration in these British is designated with the term copper. Again, different mutations in the same critical protein that has as a major biological function the degradation of the agouti signaling protein. When it's dysfunctional or not as functional, we see a transformation from black tones to red tones, or black pigment production to red pigment production, because agouti signaling protein cannot be correctly degraded. And this is, I think, the surprise for most of you. We've grown up in the cat fancy under many dogmas, in particular of genetics, of coat coloration, and this is one. I'm going to blow your minds away shortly. Another member of the pigment type switching system is orange. We've known of orange for decades. This is a sex-linked genetic information. We know that this has always been designated as the locus orange, but we still do not know what is encoded in this gene. The gene has not been, there's very aggressive work ongoing in several laboratories in the world, but there is no formal report yet. But again, conceptually, I want you to start thinking of orange as a member of the pigment type switching system, whatever this is. And in addition to the domestic cat, there's another little mammal on planet Earth with a similar pattern of sex-linked control of the pigment type, and that is the Syrian hamster. This is a tortoiseshell Syrian hamster, and the early work actually came from work conducted in the Syrian hamster. You can see in this publication in Genetics 2009 the attempts to try to map this sex-linked yellow genotype called SLY for sex-linked yellow. And where is it mapped to? The X chromosome. And these are the colorations associated with that, including the tortoiseshell. The search for orange started 2 decades ago in the cat world. There's a beautiful body of work from the Leslie Lyons lab localizing orange to the X chromosome. Some more work also from 2009 from the group at NIH led by Marilyn Menotti Raymond, again mapping it to the X chromosome, but the identity of this genetic information, what protein is encoded in the X chromosome, that has not been formally reported in the scientific literature. It's coming soon. We are crossing our fingers. It will be superb and very important information for us cat fanciers to be able to handle. So this is something we are all familiar with. It's pretty pictures, and I like to reinforce the concept what we have just learned. This is the look in domestic cats resulting from the operations of this major player in the pigment type switching system that for now we call orange. And in the cat fancy, we have recognized only 2 variants, a dominant one and a recessive one. The tortoiseshell coloration that we're all very familiar with, the majority of tortoiseshells are, of course, females. This is because this information is encoded in the X chromosome. So a dominant XOXO female or an XOY male will be pheomelanistic. The cells in the body of this animal will only be producing red pigment, and you all know that this happens from birth, whereas heterozygote females, whereas homozygote females for the recessive form or males that carry the recessive form of the gene in the X chromosome are going to be eumelanistic. In the heterozygote individuals, males or females, we will see orange patches when the inactivated X chromosome is the one carrying the recessive form, and we are going to see black patches when the inactivated X chromosome is the one carrying the dominant form of the gene. So this is one member of the pigment type switching system you've known for years, but never thought about being a pigment type switching controller. And as I said, there are others. These other players have not been identified formally in the domestic cat, but for sure they have been formally identified for other mammals. I'm going to introduce you to two of them that very likely will become players of growing interest in the cat fancy. One is called attracting, the other one is called mahogunin. And as you can see in the cartoons, they are proteins that are associated with the receptor, with MC1R, with a lock. These are proteins that control the activity of the receptor and, not surprisingly, control coloration. So I'm going to show you examples of mouse that are mutants for attracting and mahogunin. And sure enough, what you see is reddish tones in the coat when the wild type looks like this. So we see a switch again from black pigment to red pigment, different abundance, different distribution, but conceptually this is the same. So conceptually, these are members of the pigment type switching system. So,
Adriana, I'm sorry, I'm just going to interrupt you. Christine, there is a person who is not muted and it's really disturbing for everybody. Christine, can you please mute yourself? Yes, thank you so much. Thank you. Adriana, continue.
Thank you, no problem. So conceptually, if you had to clip a slide from my talk today, this is the one so far. So conceptually, you need to start thinking about coloration being the result of the control of multiple players that are operating together. So it's the combined genotype, the combined combination of variants at all of these loci that defines the genetic basis of any coloration. So whether it's black or whether it's red, and whether it looks slightly brownish or more yellowish or more oranges, it's all of these players. Some of those we know. There are several that we still do not know about, but you do need to acknowledge that these genes all act together. Agouti, melanocortin receptor 1 in the extension locus, corin, attracting, mahogunin, and orange are the ones we know about so far. So, sorry about the technological issues. We're going to go directly into group 2 now. Group 2 is a group of genes that encode enzymes that regulate the biosynthesis of melanin, encodes enzymes in that biosynthetic pathway that I showed you about. And you know all of them. It's just that probably what you didn't know is what they do, what their function is. The symbols, the colors in cats resulting from these mutations, you probably have known for a while, but what they do, what exactly they control, and in what group they would be placed, hopefully you will incorporate in the class today. So this is the pathway. We're going to talk about these genes: tyrosinase, topachrome tautomerase, tyrosinase related protein, and others. So this is the best known allelic series or collections of variants for what gene, what protein. The protein is this absolutely critical enzyme in the pathway of pigment production. This is the enzyme that converts the amino acid tyrosine in the next precursor in the biosynthetic pathway and in the next as well. And variations in this coding region, this true allelic series, because there are several variants known, is responsible for the uniform coloration or the progressively pointed coloration characteristic of the Burmese, we call it in the fancy sepia, pointed, or the combination of both in heterozygote individuals, mink. There's another slightly new variant known for this locus. It's a tyrosinase variant. Pay attention to how it's designated in scientific peer-reviewed literature. This is MOCA, discovered for Burmese individuals, and this is the look. And there are definitely heterozygote individuals that display also intermediate coloration. Also notice that some of these coloration result in intermediate colorations in the eye, also in the mocha, and hopefully you'll learn a little more about this in the talk about eye color. Very important, many of you have seen this, and it's very important to at least incorporate this concept. The mechanistic reasons are not necessarily fully understood, but it is known that some lines of pointed cats with no distinction of breed in individuals with a genotype CS or pointed cats, there is an associated deformity of the optic chiasm that results in squint or crossed eyes. For those of you working with pointed colorations in your breeds, very important to select for individuals that are not cross-dyed, because there may be some variation or associated variation in genes that are inherited together that produce this phenotype. So we're not going to go into the break. We're going to, I'm happy to stay a little longer and take questions at the end if it's okay with you, Petra.
Absolutely. I think that would be great because.
Okay, so we're going to describe the players in group 3. These are the operators that control the shape of the melanin granule and the way the melanin granules, the pigment granules, are packed into the hair shaft. So why does this result in changes in color? Again, I want to remind you all that color is what our eyes perceive as a result of how structures reflect light. So imagine if the structure is slightly different, the surface is rougher or smoother, light will be reflected differently. If there are more of these structures packed in a certain container, like the hair shaft, more packed, less packed, our eyes are going to see different color or coloration, even if the pigment is the same. So here are the examples. So the typical example is what we see in the locus B. And the locus B encodes one of these particular enzymes rapidly. This one, oops, this one. And we have known, we are very comfortable recognizing black, chocolate, cinnamon, the solid colorations in domestic cats. They are all the result of variation in this locus designated locus B. Under the control of this gene, the shape of the melanin granules is different. The shape of the eumelanin granules is different. So black is the autosomal dominant. Autosomal means that the gene is encoded in non-sex chromosomes. Chocolate is a recessive form of this gene. And cinnamon is another variant, also recessive to black. The other locus encoding melanophilin. Melanophilin is a different protein, has a different function. It controls how the granules are exported and distributed into the hair shaft. Literally, more packed or more relaxed, more spaced. And in those spaces, there's nothing, there's air. I'm going to show you pictures of hairs. Under the control of melanophilin, we recognize two major variants of this gene, the dominant D, capital D, and the recessive lowercase d. This encodes uneven distribution or clumping of the pigment granules. We see what we refer to or describe as dilution of color. So from black to blue, from chocolate to lilac, from cinnamon to fawn, and from red to cream. So here are the hairs. The pigment shapes, the pigment granules are identical. In the black hair, the shafts are fully packed. In the blue hair, under the control of melanophilin, the granules are more scattered. And our eyes see a different color. The light is reflected differently by these structures. The same for the chocolate, fully packed, more scattered. Same for cinnamon, fully packed, more scattered. What about this mysterious locus I from inhibition? We still do not know formally what the locus I encodes. We know that it encodes a dominant form of the protein that is encoded that results in inhibition of pigmentation. So that means the pigment is only deposited at the tip of the hairs with different level of magnitude or expression of this trait. So there's inhibition of transport or export or normal export into the hair shaft. And the result of this genetic control is this absolutely mind-blowing, beautiful smoke colorations or shaded colorations and the corresponding silver colorations when the individual is agouti. We're going to go into group 4. Group 4 is another territory where we have some recent news in the domestic cat universe. We're going to look at the genetic control of maturation, proliferation or multiplication, and distribution of the pigment factories, of the melanocytes, that again, remember, start their journey and their developmental biology processes at the top of the head in a place called the neural crest. So we're going to look at another key log system that is a major regulation of pigmentation in mammals. But like every single other system you heard about and will hear about, please acknowledge that other fundamental biological functions are also controlled by these key log systems. In the case of this new one that I will introduce you to, any event of cell survival and proliferation, hematopoiesis, that means the genesis of blood cells, gametogenesis, that is the genesis of oocytes and spermatozoids, are also impacted. So, of course, melanogenesis, or the production of pigment. This is the KIT and KIT ligand system, key log system. KIT is the receptor or the log, and KIT ligand, also known as stem cell factor or steal factor, is the key. So, in this process involved in melanogenesis or melanin production, the interplay between the log or c-KIT and the ligand, stem cell factor, the key, will result in the following. There will be a control of the migration from the neural crest to the terminal distribution or final distribution or of that pigment producing cell. There will be a control of survival of this cell during development under some of these genetic controls that I'm going to tell you about. The melanocytes don't survive. They die or they lose function, and that means there is no melanin production. There would be also control on the proliferation of these cells, and there will also be a control of the activity of these cells. And I introduce it here because I will not have time to talk much about it, but the hair graying, the loss of pigment in the hair shaft, is a reflection of a reduction of the activity of the factory. If the factory doesn't produce enough product, the number of pigment granules in the hair will be less, or the volume of production will be less. So, very important that you understand how the migration of these melanocytes is programmed. Remember, I told you these cells originate in the neural crest and populate over the development of the embryo, the individual, populate under the control of this system of key and lock. They will populate areas in the eye, areas in the skin and hair. They will populate a specific area in the inner ear required for function. And in this particular case, the function is a critical one, and that is the hearing function. And they will populate also regions of the brain that we are not going to discuss today. So, as you can see, when the migration is complete, we see embryos of uniform color. The whole body is populated by melanin producing cells, whereas in a bicolor embryo, this migration is controlled. Only a few cells get to the tail area, and a lot of cells stay in the head or apical area. So these are terminologies that many of you may not be familiar, but the migration is controlled in a specific fashion. The directions of migration are head to tail, in technical terms, apical, caudal, head to tail, and also dorsoventral or back to abdomen, from the spine to the belly. So definitely the genetic basis of the control of white spotting or particolor pigmentation in cats has been under study for many, many years. This is literature and reports of research findings from 2010, 2014. There's still quite a bit of work ongoing in this territory, and I'll tell you more about it shortly. But as a scientist, I think it's fundamental to recognize the work of our colleagues that has been instrumental to our ability to test, to classify, and to designate colorations in purebred cats. So this is the way the research in these publications was reported. Essentially, these authors identified the genetic basis of white spotting as mutations in the KIT receptor, in the c-KIT coding region. Again, this is the log. Different mutations resulting in different colorations, full coloration or colorations that include white in different magnitudes. You can see it here. has gene sequence data. I'm going to summarize the conclusions for you because this is ultimately what it's important for us in the cat fancy. So essentially, this paper was absolutely revolutionary because this paper, this body of work, 2 papers, immediately eradicated the use of the term S that designated piebald spotting. We no longer use S. We now adopted the universal genetic nomenclature W to designate white spotting. So the name of the locus is not white, it's white spotting. And the findings unequivocally identified one single coding region that controls solid white and white spotting phenotypes or particolor phenotypes. And that is localized to the KIT receptor gene. And this body of work identified 3 variants, 3 alleles, dominant, capital W, and 2 recessive ones, WS and just plain W. Very important, this is mechanistic. For those of you who are interested in accessing the literature, please communicate with Petra and I will be happy to facilitate access to the research papers for those of you who can and want to read more. So the mutations have been localized to different mutations in this particular coding region. And these are different kinds of mutations. These are actually insertions of retroviral sequences. The insertion of retroviral sequences, by the way, happen to be major causes of genetic variability in any creature living on planet Earth, from humans to insects. So what are the possible genotypes or genetic makeups at the white locus, and what are the resulting coloration phenotypes? When we have at least 1 copy of the dominant gene, the cat is white or solid white. When we have 2 copies of this variant WS, we see different magnitudes of white spotting. When we have 1 copy of WS and 1 copy of white, we see a completely different distribution of white with higher dominance or higher abundance of pigmented areas. And when we have 2 copies of the W variant, we have no white, solid pigmentation. Very important, and that is the concept of heterochromia, or in the fancy, odd eyes. This is a eye coloration phenotype that is frequently associated with white spotting, coat coloration phenotypes. And this is a coloration phenotype that has pathological implications, and that includes deafness, lack of hearing function. Why? As I mentioned a few minutes ago, the production of pigment in the inner ear, remember the inner ear is one of the areas of the body that gets colonized by melanocytes. Now we know that the production of pigment is required for the hearing function. So every time, especially for those of you in the audience that are veterinarians that see cats in the clinic, many times the consultation for cats with heterochromia is that the owner thinks the cat doesn't hear well. So sure enough, hearing should be tested. There are beautiful, very elegant studies published reporting impaired hearing function in cats with odd eyes or eye heterochromia. This is what we know. This is important because it's our responsibility to understand that these colorations that are very pretty do have occasionally pathological implications. The white spotting coloration in the Burman, definitely known for decades to be the result of a different genetic control, characteristic of Burmans and mitted ragdolls, designated with the letter G, and this is because the genetic basis was very clearly identified as different from white spotting. This happens to be the result of mutations in the KIT, in the receptor as well. Very nice work published, reported by the group of Montang and colleagues in the proceedings of the National Academy of Sciences in 2014, essentially reporting the sequence of the gene and the location of the mutation in the KIT coding region that is different from white spotting. So very important, and we are still in this group of genes that hopefully you have realized how important they are and how some of them need to be watched carefully because there are pathological implications for some of these beautiful colorations in cats. There are other genes involved in the control of melanocyte proliferation, development, and pigment-producing activity. This is the melanocyte, and we have already learned about this key log pair. We just learned about this key log pair, KIT and stem cell factor or KIT ligand. We're going to learn about other players. These are transcription factors. There are many, as you can see inside that nucleus of the melanocyte. We are not going to discuss them all, but these two transcription factors, MITF and PAX3, we are going to discuss because it's clear that mutations in these genes are resulting in colorations that are rapidly infiltrating the cat fancy. I'm going to continue showing you the mouse because I think understanding what happens in mice will help us and should help us. It is absolutely clear that a lot of the findings from other mammals can be extrapolated to mice, and hopefully you'll understand that a lot of the discoveries made for other mammals are serving as major sources of clues for feline genetics researchers to search for the genetic basis of different colorations in cats. So these are mice, and all of these colorations that are particolors or white spotting colorations in these particular cases are results of mutations in the transcription factors MITF and PAX3. So now I'm going to give you a very important clue, and that is the name of the gene called MITF. This gene is called microphthalmia gene. And this exotic term that many of you may find difficult to pronounce literally means small eyes. So this mouse, maybe it's a little hard to see in the picture, but this eye doesn't have a normal size. This is a pathologically small eye. So this gene is called microphthalmia transcription factor, MITF. Now, look at this, very cool. Many of you have seen, I love this coloration in horses. I'm going to show you something hopefully eye-opening, and that is that these colorations are also the result of mutations in these two transcription factors, MITF and PAX3, They cause what horse people call splashed white and other white spotting phenotypes in horses. So don't worry about the legend. Just look at the designation of the mutations. It doesn't matter. Conceptually, I want you to see the diversity of code phenotypes that can result from mutations here in these transcription factors. In general, this is more localized white spotting. Can you see? It's not the whole body like with W. It's more localized. If you think about it, hopefully you're making the connection to the extremities, or if you want, for the terminology that we use in the cat world, to the points, extremities, colder areas of the body. But hopefully you're going to see this, and I hope the magnification of the slide is good enough. The eye of this horse, I apologize, couldn't make it any bigger. This eye is blue. The eye of this little baby horse is blue. We are lucky to have even more information from mutations on the MITF gene transcription factor. The mutations in MITF are responsible for the white spotting colorations in Newfoundlands. They are also responsible for the white spotting coloration in Dalmatians. And many times in Dalmatians, there is pathological condition. And look at the eye color of this Dalmatian. Now, hopefully you recognize this species of mammals. These are humans, everybody. And hopefully I'm showing you humans with very distinct pigmentation of their skin. So you cannot say, oh, I'm seeing non-pigmented eyes because the human is white Caucasian. I'm showing you a diversity of skin colorations in humans with common features, blue eyes, different ethnicities, all with the same characteristics. This is a very obvious example of white spotting. I'm sure everybody can see it, same as this one. Probably it's a little bit hard to see, but there's white spotting here in the eyebrow. And there may be a little here. In this boy, it's a little hard to see. What is this condition associated with mutations in MITF in humans? This is called Waardenburg syndrome type 2. So in addition to having hypopigmentation or lower pigmentation, lower levels of pigmentation in the skin, ocular pigmentation defects. Why? Because this eye in a normal individual should be pigmented. Please pay attention to this. There's also deafness. Why? Because in these individuals, the melanocytes in the inner ear are not functional. So this is a pathological condition, very well characterized for humans, associated with mutation in these two transcription factors. Now, back to our world, the cat fancy. There are some very rare phenotypes that have been described, reported, registered in different cat associations. The phenotype called Celeste or Celeste in Spanish. This is original material shared with me by Pascal Portelas that works for the French registry Louvre. These are blue or odd-eyed individuals that should be normally fully pigmented. As you can see, and recognizing the same pattern we saw in horses, the same pattern we saw in humans, very limited white spotting. In some of these cats, you can barely see the white spotting. Very limited white spotting. But we definitely see blue eyes, or like in this one, these two girls, heterochromia. Definitely this has been reported for Maine Coons, and this has been also reported for Bengals, heterochromia and blue eyes in individuals that are otherwise expected to have fully pigmented eyes. Research is ongoing, but it is extremely likely that these coloration phenotypes result from genetic variability in MITF or PAX3. And definitely there is ongoing research work focused on elucidating the genetic basis of these conditions, and definitely using as a platform for interrogation of the genome what we know about the mutations in these genes and their impact in coloration in other mammals. Hopefully this will blow your mind away. It's not just cats, humans, and horses that present with heterochromia. This is a defect seen, recorded in other species of mammals, as exotic as koalas from Australia, wolves, jaguars, lemurs, goats. So we humans have been exposed to this. We may have not paid enough attention, and we may not know enough about the genetic basis. But for sure, what we are obligated to acknowledge are the pathological implications. So to finish the presentation today, we're going to discuss what we know about the genetic control of tabby pattern. These are genes that definitely fit in group 4, and that's why they are presented here, because the tabby pattern is definitely the result of a very well programmed migration of melanin producing cells. And these are the basic patterns that we recognize in the fancy, classic or blotched, mackerel, spotted, and ticked. A good reminder that pattern is critical for mimicry or camouflage. If the individual has to live in the wild, it better have a pattern that allows it to disguise from predators. That's a whole function, biological function or pattern. Without a doubt, this is visually spectacular. I think we humans call it beautiful, but this has a fundamental biological function in the wild. So in the fancy, we have recognized and been very aware of a true hierarchy of pelage patterns. We have recognized the markings seen in the Abyssinian, and we know that they are sort of dominant over the spotted coloration, the mackerel coloration, and definitely the classic coloration. Very aggressive and beautiful comprehensive work from the laboratory of Marilyn Menotti Raymond, again at the National Institutes of Health, where there is a huge group interested in canine coat color genetics or canine genetics in general at the moment, and definitely where the majority of the feline genetics researchers started training and branching out to start their own lab. So these groups are the ones that contributed the body of work that has allowed us to understand the genetic basis of tabby pattern. And there are 2 different coding regions that are responsible for tabby pattern. These are essentially the conclusions, what these studies elucidated. The ticked locus TI and the tabby locus TA. So 2 are required to control tabby pattern. It is known now that the tabby locus TAs encodes a protein called TACPEP or leverin, transmembrane aminopeptidase Q, for those of you who are interested in scientific terminology. So different variations on this gene are responsible for the mackerel and the classic tabby pattern. Allelic variation is responsible for the arrangement of light and dark colored areas, or actually pattern, what we describe as pattern. These markings, like the markings of the tiger, by the way. Importantly, it's mutations in this same gene, TACPEP, that are responsible for variations in the markings in the cheetah. This is the regular, most prevalent coloration phenotype in the cheetah, but as you know, these spectacular, beautiful looking king cheetahs exist in nature. This is definitely a recessive mutation in TACPEP that results in larger pigmented areas on the body. So from these studies, it is important that we recognize two loci controlling coat markings in cats in general. Let's restrict our discussion to domestic cat. The ticked locus designated TI that determines the presence or absence of a pattern. I'll show you specific examples for you to fully understand this. And then the tabby locus, designated TA, that controls the size and shape of the marking. So again, tick locus controls the presence or absence of pattern, and the tabby locus determines the size and shape of the markings. And what we describe as modifiers, one or more additional loci that will modify that pattern further, like transforming mackerel into spotted, or even classic into larger spots that would break the pattern. So here are the examples. The agouti tabby characteristic of the Abyssinian is encoded by the variant TIA, or ticked A, dominant. And what we should describe here under the term ticked, essentially, we're in a phenotype that is characterized by the absence of pattern. No tabby markings visible. When the variant in the ticked locus is variant designated TI1, then we can see the pattern, so it does matter what is encoded in the tabby locus. In this particular case, the classic tabby or blotch tabby encoded by the recessive variant of this gene. And we are all very familiar with these looks. Of course, this is all readily visible when the individual is agouti. In the mackerel, again, if we see pattern, you need to know that the variant in the ticked locus has to be variant 1, because if it's TIA, we do not see pattern. And in this particular case, we have this dominant form of the tabby gene that will allow us to see this characteristic pattern with stripes or mackerel. Notice that because of the dominant recessive relationships between mackerel and blotch, one copy of mackerel is enough to see stripes. But again, information we've had for decades, and we've handled this beautifully without even knowing the genetic basis of tabby pattern. When the individual is heterozygote, now we know that when the individual is heterozygote for the ticked locus, one copy of A, one copy of 1, there is some visibility of the pattern. Stripes in the legs, stripes in the tail. Sometimes it's possible to speculate whether that is mackerel or that is classic based on how broad they are, but for the purpose of this discussion, that's irrelevant. What I'm showing you is heterozygosity in the tick locus and the return of visibility of the pattern, at least in some areas of the body. So when you see pattern, you know you are looking at heterozygosity in the tick locus. When you see pattern in the extremities, but not in the body, that is the case. These are spotted cats. So notice that here I'm plugging in the term modifiers. Why we call it modifiers? Because they have not been formally identified. But we know that the function of these additional player or players, more than one perhaps, is to break the pattern, to break stripes into small spots or larger markings into bigger rosettes. So, what have you learned today? I apologize again for the technical problem. I got kicked out of the internet, but hopefully I was able to deliver novel content to you to open the doors of your brains to access new information and keep seeking new information. This is a rapidly growing field. We are going to be flooded with new data very, very soon. So you've learned a basic concept, and that is that the production of pigment is a defense mechanism for protection from mutagenic UV radiation. If there are people in the audience that are involved in groups watching or trying to impact cat welfare, be prepared. In some countries, the legislation is already against the breeding of white individuals, non-pigmented individuals. This is the rationale. You have learned that there are two major key lock or ligand receptor systems and their associated signaling proteins that control the development and activity of melanocytes. The agouti protein and the receptor, and then the stem cell factor and the KIT receptor. You have also learned that the grouping is conceptually important. You've recognized groups that control enzymes that control the switching on and off of the factory and that control the proliferation, differentiation, and migration of the melanocytes and their activity in their final destinations. You have also learned about the term melanism, and you know that mutations in both the agouti locus that encodes the key or the receptor locus MC1R that encodes the lock, that mutations in both sides can result in melanism. And you have also learned that there are at least two identified, one or more not identified to the present, that control the tabby pattern phenotype. Ticked, tabby, and the modifiers. Hopefully, you have also learned and deposited this critical information in your toolbox, and that is that there are genes implicated in colorations in cats and other mammals, including humans, that do have pathological implications. And that is our responsibility to better understand this situation, because we are responsible for not only the quality, but the health of our companion cats, and producing animals that will have health issues or functional defects. I personally think it's completely unethical. So breeding programs that may be operating with some of these genetic bases need to be responsible, need to have the information, need to have the ability to make informed decisions in breeding. Definitely, hopefully you have learned about candidate genes that are actually being targeted as we speak and need to be continuously targeted for further research, not only to understand or elucidate the genetic basis of some of these novel white spotting phenotypes. We need to understand the pathological implications, and if there are pathological implications in the domestic cat. Without research, this is impossible. Without research, we will not have access to testing panels. So research is fundamental, and any of the groups that are partnering with research laboratories are actually making a lot happening for the cat fancy. We are beginning to receive a lot of return for our investment, for our support to feline genetics research. Hopefully, and the last conclusion for the talk is that pretty or novel looking is not always functional. And again, this lecture is not on ethics, but again, ethics in breeding is something we have to continue watching and monitoring. We have to acknowledge that many of the traits that we humans have selected to perpetuate in domestic animals of many kinds result from loss of function, and loss of function should be watched carefully. Why? Because our cats, the cats we breed, are not under the very critical, dramatic impact of natural selection, the survival of the fittest. We decide who survives, who breeds. And many times what we are propagating is detrimental to the individuals that present with those characteristics. So with that, I thank you for your attention, and I'm very happy to take questions.
Hello, thank you so much, Adriana. I absolutely loved it. I've learned so much. And there are actually a couple of questions if we still have time to answer those. So there is one, if the white or the gloving in Burman cats, sacred Burman cats and mithed ragdolls is encoded by the same gene, and if yes, why is the pattern different in those breeds?
So that's an interesting question, and the answer it is technically it is the same gene, it is the same coding region. Hopefully it will be helpful if I tell you that the coding regions are huge. And in genetics, especially for the sake of testing, developing panel testing or conducting more handicrafted genetic testing by polymerase chain reaction based approaches, different locations in the gene are equivalent to having two different genes. These are two different locations in the gene, so technically two different loci. The coding region is the same. This is the same gigantic kit, receptor kit. So I think operationally it's okay to describe this picture as two different genes. And that's why even before the genetic basis were understood, the designation of these genetic bases use different letters. G for gloving and originally S, now W for white spotting.
Okay. Thank you so much for answering this question. There's actually another one about Bengals. So if somebody has the goal to breed Bengals with the coloration of ocelots or margais, are cats that appear to express the corin going to be help or hinder with the progression? Often Bengals with counter shading appear to have corin, but this is not the same counter shading color as the few have black spotting. And I'm not sure if mixing Korean Bengals with counter shading with non-Korean Bengals will affect progression towards my goal.
Well, that's a very complex question. I hope I can answer it correctly. The first thing I'm going to say is that unless your cat or cats have been formally tested for Korean mutations, and that requires sequencing, gene sequencing, you do not know if your coloration is a result of a Korean mutation. That's the first thing I want to say. I also remember hearing in the question something about the coloration of the margay. So what I want to say in this context is that if there's a new species involved in your breeding program, in this case, the margay, be prepared to have variants on these key player genes that we discussed as part of the pigment type switch system that may be unique variants. And I honestly don't know enough. I know laboratories like Chris Kalin in Stanford. He's interested in looking at these coding regions for other Felidae. I am not aware of any particular findings for margay or other wildcats, but it's very likely, like we got the agouti signaling protein from the Asian leopard cat. It is likely that the variant that is encoded in the genome of margays is different, and we do not know how these variants operate in the context of what is, depending on the breeding program, mostly a domestic cat genome. We simply, I don't think we have enough information to comment or to advise on strategies in breeding program. My advice, and that is because my knowledge is fairly limited, just to be so specific to the Bengal breed, there are many chat groups, many people with a lot of experience, many people that have been breeding and looking at the result of breedings with cats that have their genomes, their complete genome sequenced. So those people may be able to provide better advice.
Okay. A lot of thanks, and I think a lot of people really enjoy your talk, Adriana, so thank you again. And there is one question actually, which it's a new topic for discussion. It's been quite on the topic. It's for the blue-eyed Maine Coons with little to no white. So the question is, there has been much discussion about the blue eyes in the little to no white Maine Coons as being from illegal outcrosses or breeding of cats with recessive genes that present health issues. And what are your thoughts on this?
Well, these are my thoughts. I think we need more research. I think it should be fundamental that, especially after this talk, you have your antennas up, and every time you see blue eyes, especially from blue eyes with unusual, very, very dense and unique, distinct tone of blue, that your antennas are up on alert, especially if there are no obvious white areas in the body. Unfortunately, I am not aware that there is active genetic testing targeting MITF or PAX3. I know there are laboratories working on this, but I do not know if the tests are offered. And again, because we are a multidisciplinary group of people delivering this talk, this collection of talks, very important that you understand that to make things better in the cat fancy, we need partnerships. We need scientists partnering with veterinarians and breeders. So my take on this is that it's fundamental to assess if the pretty eye coloration is associated with a detectable impairment in hearing. If you have an impairment in hearing, you know that even if you don't know the genetic basis, there's something pathological about the coloration of the cat you have. Again, I think the simplest way of putting it is pretty is not always functional, and that's where our ability to make the right call on whether what we're doing is ethical or not is something, I guess, between ourselves and the mirror. But my take, how I would handle a breeding program along those lines, is to make sure that the odd-eyed phenotypes, the heterochromias, or the blue eyes in cats that have no visible or minimal visible white spotting, that those are monitored functionally. And the obvious one would be assessment of hearing function. The other thing, Petra, if you don't mind this comment, without testing, the registries will not have the ability to place those cats based on the coloration phenotype, because based on the coloration phenotype, I mean, we may have the ability to have an educated guess and may have the ability to segregate those with minimal or no visible white into a different group and flag them. The typical odd-eyed bicolor cats, those, I think we also know that some of those have hearing impairments. So again, monitoring for health, I think it's absolutely basic. With these novel colors, we are still running a little behind, very limited with our tools to assess or determine the exact genetic basis of the coloration we are looking at. So, and that's the reason why some of these coloration phenotypes have infiltrated the pedigrees, because there's no way to track them. Essentially, the people that should be tasked with the responsibility to track these animals are the breeders. And that's why you breeders are the target for our efforts in continuing education.
Okay. Thank you, Adriana. I couldn't agree more. I definitely think it's always great to have new colors and new breeds and everything, but I think we really need to focus on having healthy cats and continue really focusing on the health and welfare of the cats as well. So thank you, everybody, for taking part in today's conference. We will meet again tomorrow at 8 a.m. Mountain Time, 4 p.m. Central European Time. Hopefully this time without any technical issues, because now we've run really over time. So I'm hoping the fact that almost everybody is still here means that people have been enjoying it. So thank you so much, everybody. Thank you to all the amazing speakers, Danielle, Adriana, and Sarah. And also thank you to our sponsors, Royal Canyon Merck, and our media partner, Every Cat Health Foundation. And we will see you tomorrow morning or afternoon if you're in Europe.
Thank you, Petra. This was great. Thank you.
Thank you. Thank you, everybody. See you tomorrow.